content. Introduction, background and motivation, discuss about what is background scenario in blood counting, and problem situation, discuss about problems in blood counting, and in others works discuss about what are the techniques available in using image processing, how to segment those cells, and hypothesis, technological adapt, methodological approach, implementation and result, conclusion and reference. So if you have dengue or when you attend to hospital or when you meet a doctor, basically they tell you to bring a blood report. Or so maybe it's FBC or full blood report or WBC white cell count. These blood reports are created based on microscopic pictures. So basically there are two techniques, manual technique and automated machine technique, like hematology analyzer. So but also we can apply image processing for cell counting. So many researchers are use thresholding based technique for detecting cells. So when you go into problem, these automated machines are very fast and high accurate, but the problem is one unit is more than 3,500,000 rupees more than. So and also it is not this less number of units are available in our country, and also these machines are not operating properly sometimes. Most of the time lab technicians use manual technique, and the problem in manual technique is time increasing and inaccurate process. It is also depend on the experience of the lab technician. To create a blood film, it's take approximately 30 minutes. For counting, it's going for another 20 minutes. And if you have dengue, you have to calculate blood reports every 2 hours. So when you go in using a manual technique, it is time wasting and some problems. So in others works, there are some many research going on blood segmentation using image processing. Most interesting and important thing in our research is blood segmentation, because actually human brain can understand objects, but in computers they can't understand those kind of things. So we have to use some mechanism, we have to create some algorithm to identify those kind of things. So I will skip these others works and going for summarization table because we have less of time. And when you go to summarization table, the basically segmentation, cell segmentation using image processing, there can be categorized into 4 types. Thresholding based, using morphological operation, segmentation using watershed algorithm, and cell fitting using edge detection. When you talking about thresholding based, in most biological related images, thresholding like X-ray, MRI images are used also algorithm for thresholding. And it is very good for cell segmentation. And if you thinking about morphological operation, we also can segment cells using this operation, but to filling holes, many authors propose this morphological operation like opening filter, but it is best for a particular image. But when we apply different image, we cannot use this morphological operation. And watershed algorithm have same problem in overfitting or underfitting errors, and circle fitting using edge detection also good for cell segmentation. My hypothesis, computer software using image processing can be used for blood segmentation, and combination of thresholding operation with edge detection and ellipse fitting algorithm can be applied for blood segmentation, and using purpose algorithm can be used to reduce the time delay for counting the cells in manual technique. So these are the technologies I use OpenCV used for image processing, and Python for program the algorithm and our system. And the system have a little bit database part for store patient data. So we use SQLite database, and scikit-learn also use Qt used for design interfaces. And totally this technologies are open source technologies we use. In the methodological approach, I will discuss about our proposed algorithm. Basically, algorithm have two stages: pre-processing stage and post-processing stage. I will explain in pre-processing stage. First of all, we have to take blood image, microscopic blood image from microscope, and then insert into our proposed system. Then we have to apply noise remove, because if you use low quality camera, image can be have some noises. So we have to remove those noises using noise remove method. Then you have to split RGB component, because if you going to detect red cells, so you have to use green component. So we split the image to RGB component, and then we take green component channel and then apply thresholding. In here you can see holes in the image after thresholding. We use in here also thresholding technique, and before going to post-processing stage, we have to fill those holes. So in here we not use morphological operations. For that we apply masking method. Using masking method, we can fill holes and then go for post-processing state. In post-processing state, we have to get edges. So we apply in our algorithm canny edge detector to detect edges. Then apply contour detection for segment cells. Assume this is 213 cell, and after contour detection, you can see some small dots in image. So using contour points, we can segment this image, this cell. So before that, we have to reduce these contour points. So we apply polygon approximation. After polygon approximation, we have approximated shape of the cell and the less number of contour points. Then ellipse fitting. If you have problem how to count the cell, so this is a basic core part of the algorithm. In our algorithm we declare some three arrays for store ellipse area, and based on the ellipse area we segment the cells. And first of all we calculate the mean of the ellipse using ellipse area list, and identify single cells, and also identify touching cells. Then we for segment the touching cells we apply concave point. We have to find concave point. We already have polygon approximation point, so we wrote some function to calculate the concave points and check this function go to the pointers and check whether its particular point is concave point or convex point. If it is concave point, then apply touching cell splitting. For that again we apply ellipse fitting. So in here you can see using those concave points the circle will split. And this is original image how we split. You can see blue dots are showing the polygon approximation point, and red dots showing the concave point. Using after ellipse fitting they are segmented. And finally we can add those count to the single cell array in previous line showing the code, and using array length we can count the RGC count. This is my implementations, and we have some login based to into system, and system has some lot of functionalities, because the lab technicians have to work with a lot of workload, so we have to reduce lot of workload. So system have some shorter functionalities, and this is about results. So EN means estimated number of cells counted by our proposed algorithm. And conclusion is our proposed algorithm can identify red cells and count them within less than 1 minute, and also it has more than 85% accuracy for cell detection and segmentation. However, this accuracy depends on the image quality and its brightness. These are reference. And I would like to thank for Mr. Pathirage, my supervisor, and Mr. Kulasekara, my co-supervisor, and also I would like to thank Mr. Kulasekara for teaching about image processing and its application. Thank you.